Okay, so we're going to pass to the next speaker. So it is my great pleasure to introduce you Dr. Mirjana Efremova. She leads a lab at Bart's Cancer Institute focused on investigating non-genetic mechanisms of therapy resistance and metastasis in colorectal cancer. And Dr. Efremova developed a cell-cell communication statistical framework, CellPhoneDB, and also brings deep expertise in cancer evolution and single-cell transcriptomics. So please join me in welcoming her. So the floor is yours.
Thank you very much, and thank you for the invitation and the opportunity to present our research here. Mirjana already gave us a really beautiful introduction of phenotypic heterogeneity and plasticity, and I will follow up on this, focusing on metastasis specifically in colorectal cancer. So as we heard from before, cancers comprise a heterogeneous collection of cells that each have diverse genetic and phenotypic variabilities, and this enables them to have different roles in tumor progression, metastasis, and therapy resistance. And in addition to genetic variation and genomic instability, this heterogeneity also derives from phenotypic heterogeneity, which is due to the ability of the cells to transition between different states so that they can respond to external signals and that they can easily adapt to different environments, for example, those encountered during metastasis or under therapy pressure. And this ability is called plasticity, and what we're interested in the lab is looking at both cell intrinsic factors, for example, the epigenetic regulatory factors, as well as the signals from the microenvironment that enable this phenotypic heterogeneity in metastasis and therapy resistance. So, as also Mirjana mentioned, there are different forms of plasticity. So, for example, the cells from a differentiated cell state can de-differentiate or revert back to a stem cell state, can even go further down the developmental trajectory and start to look more like a fetal state. But then also there can be a process of transdifferentiation where a differentiated cell state can transition into a different type of cell state, and the most common example is epithelial to mesenchymal transition. So in the lab, we are focusing on colorectal cancer. Colorectal cancer is the third most common cancer globally, and it's the second leading cause of cancer-related death, and this is mostly due to the high relapse rate. So the 5-year survivor for early stages is quite good. However, for metastatic disease, less than 1 in 5 patients will survive more than 5 years. And most of the colorectal cancers metastasize in the liver, and so up to half of patients will present with metastasis, and then up to 80% will relapse after resection. And in terms of treatment, unfortunately, most of the metastatic colorectal cancers are unresectable, so they are usually given chemotherapy. It's usually a combination of different drugs. However, around 40% of advanced cases will not respond, and we are currently unable to predict which patients will respond. So colorectal cancer recapitulates the hierarchy of the normal colon, which is maintained by these stem cells that sit at the bottom of the crypt. They are LGR5 positive stem cells, and that they give rise to the progenitors, which are called transit amplifying progenitors. These are proliferating cells that then can differentiate into the different subtypes of cells in the colon. So we have the absorptive colonocytes, and we have the different types of secretory cells, which are the tuft cells and the enteroendocrine and goblet cells, and they perform different functions. And so the stem cells are dependent on Wnt signaling, whereas the differentiated cells on BMP signaling. So, but however, there is a high degree of plasticity, both in the healthy intestine, and of course this is kind of hijacked in cancer. So studies have used lineage tracing to show that if you damage the stem cell pool by radiation, for example, or infection, then the differentiated cells can revert back and become stem cells and repopulate this niche. And the same is true in cancer, so this presents a really big challenge in terms of treatment. And we know now that this is really important in metastasis also, because there have been many genomic studies with big cohorts of patients where they have looked between matched primary and liver metastasis, and they haven't really found a common genetic reason or cause. And we also think that, at least from the studies, it seems like metastases are seeded quite early. So all of this indicates that plasticity plays a role. But two important studies in mouse models have really proven this by using, again, lineage tracing techniques. They've looked into the cells that disseminate to the liver, and they've shown that those cells, contrary to what was taught before, they are not stem cells, they are LGF5 negative cells. And they start to upregulate signatures of regeneration, or the same signature that is present when there is injury in the intestine. Specifically upregulating this marker MP1. However, when they get to the liver, then in order to successfully grow the metastasis, they need to revert back to LGF5 positive stem cells, and then they can recreate the hierarchy there and regrow the tumor. So underlying all of this process of plasticity are these so-called regenerative or revival cells. And they are described, as I mentioned, as a transient state, both in healthy intestine, for example, when there is certain damage, radiation, or infection, but also the same signature have been described in cancer and in these pro-metastatic cells. And interestingly, they seem to upregulate also signature of fetal intestine, which is derived. So in this study, they grew organoids both from fetal and adult small intestine, and then they compared the signature with these regenerative or revival cells. And they have been described with a different name in the field, either revival or oncofetal or regenerative, but they refer to the same cell state. And finally, this process of plasticity is also important in therapy resistance. Again, genomic studies haven't found a shared cause or genetic mutation. So the assumption is that either all cells or subpopulations of cells is capable of entering this drug-tolerant state through process, again, of plasticity, which is a slow cycling state. And then post-treatment, the tumor can regrow again. And our preliminary data, but also other studies, have shown that, again, this converges into this regenerative fetal-like state that is, again, transiently upregulated during this process. So it seems to be a common state that appears or shows in the intestine when there is any kind of stress or injury. So, however, we don't know what are the regulatory factors of these different phenotypic states, both intrinsic factors, but also the signals from the microenvironment that could maintain or push the cells into these different phenotypic states. So our approach was to use single-cell multiomics and spatial transcriptomics data from patient samples, both from primary colorectal cancer and liver metastases. And they were both derived from our own tissue bank, as well as publicly available samples, so that we can characterize the different phenotypic states in primary and in metastatic colorectal cancer, derive their transcriptional regulators, as well as understand their spatial position, and all understand which of the cells they interact with them. So to start with, we first focus on primary colorectal cancer. Here we use single-cell RNA transcriptomics data. This is mostly from public studies, where we collected around 100 samples. So each dot on the plot represents a cell. It's colored by its cluster, and each cluster is annotated based on our own annotation or known markers in the literature. And so, as expected, we detect all of the cells that are present in the normal colon. So we see stem cells, we see the transit-amplifying progenitors, we also see the differentiated colonocytes and the different types of secretory cells, so enteroendocrine, goblet, and tuft cells. But in addition to this, we also find cancer-specific states, which we don't find in the normal colon. So one of these is a state we call stem notum. So it looks like a stem state, but it also upregulates Wnt inhibitors such as notum and other genes, other Wnt inhibitors. And this is relevant because in a mouse model, they've shown that cells that have APC mutations, APC is one of the most common mutations in colorectal cancer, and they've shown that cells with those kind of mutations, they can outcompete the wild-type stem cells in the crypt by secreting the Wnt inhibitors, because the wild-type cells depend on Wnt inhibitors to exist. Otherwise, they will push them into to differentiate, and that's how they outcompete them. In addition to that, we also find these regenerative cells that I mentioned during the introduction, and we find two different subtypes of them. With one of them, we call inflammatory regenerative cells. We call them REX and IREX because it shows upregulations of signatures of regeneration. And between them, we find a cell state that we call it intermediate because it sits just between the stem nodum and regenerative cells and indicates potential transitions or a more plastic state. So focusing on this regenerative state, we see that they upregulate these signatures of regeneration or revival that have been described in the literature. They also upregulate the fetal intestinal signature as expected, and YAP signaling. YAP signaling have been shown to be very important for transitions into this regenerative state. And they very closely resemble the metastasis-initiating cells that were described in mouse models. And a subset of them also shows signatures of inflammation. They lack classical markers of epithelial to mesenchymal transcription factors. However, they show markers of partial epithelial to mesenchymal transition. And also, they upregulate two known signatures of worse prognosis that are known in colorectal cancer. When we looked at all of the patients, we saw that there is a lot of heterogeneity in terms of the composition of cell states. So some of the samples would have a very large proportion of these regenerative cells, and some would have much smaller proportion. So to look into this more, we then divided the samples based on their transcriptomic subtype. So the most common transcriptomic subtypes, there are many different ones in colorectal cancer, but the most commonly used in the field are so-called consensus molecular subtypes, where people have used transcriptional data to divide them into 4 different subtypes, CMS 1, 2, 3, and 4. So CMS1 is enriched by samples with microsatellite instability. These are samples that have lots of mutations because of this damage or issues with the microsatellite instability. And they also are characterized by high immune infiltration. And those are the samples that best respond to immune checkpoint inhibition. CMS2 is the subtype with more canonical or stem-like phenotype with a lot of Wnt signaling. CMS3 is more metabolic subtype enriched with secretory cells such as goblet cells. And CMS4 is the one that is characterized by the worst prognosis and is also characterized by a lot of stromal infiltration. So reassuringly for us, the stem cells were enriched mostly in this CMS2 subtype as expected, and the goblet cells were more in the CMS3. However, the regenerative cells, we saw that they're enriched in the CMS1 and CMS4, which is a bit contradictory because one of them has the best prognosis and one of them has the worst prognosis. However, this tells us that their phenotype is potentially driven by the microenvironment. So then we wanted to look what happens in liver metastasis. So this was all primary cancer up until this point. So for this, we generated our own data and we used multiome, so where we got gene expression and chromatin accessibility from the same cell in order to be able to also infer some regulatory elements that could be important for driving these cell states. And first, surprisingly, we saw that we can find the exactly same cell states in liver metastasis. So it seems that whichever is the prometastatic or disseminating cell state, they do recreate the same hierarchy at liver metastasis. And the cells also express the same signatures. To validate this, we also used a data set where they used matched, because our samples were not matched, we used matched primary liver metastasis, and we showed that, again, the cell states are reestablished in liver metastasis. But since now we also have paired RNA, so paired gene expression and chromatin accessibility from the same cell, this enabled us to look more into what are the regulatory factors. So for this, we turned into the open chromatin landscape, and we first wanted to predict enhancers by correlating expression of genes with the accessibility of distal elements within certain distance. And we were able to find around 1,400 putative enhancers. When we compared them with ChIP-seq data obtained from a colorectal cancer organoid, we had a very big overlap showing the validity of the approach. And then we clustered these potential enhancers based on their accessibility. And what we could see is that they nicely clustered into different clusters that had, that when we mapped this to the gene expression also, we could see that they show that they are reflecting the gene expression of markers for the specific cell types or cell states. So we could see one cluster responding to the stem node, one to the stem cells, then to the colonocytes, and to the secretory cells, so suggesting that they are very much epigenetically regulated. And for us, the most interesting one was, of course, these two clusters that were corresponding to the regenerative cells. So first we looked at pathway analysis of the genes that are potentially regulated by these enhancers. And some of the terms were, of course, expected: angiogenesis, epithelial to mesenchymal transition, hypoxia. But we also saw, for example, upregulation of MAP kinase signaling pathway. I'll come back to that a bit later in the talk. But then, as a comparison, for example, we saw Wnt signaling in the cluster that represented the stem cells, so it was quite reassuring. Then we looked at which transcription factor motifs are enriched in, in the open regions in these clusters, and what we could see is that subunits of AP1 and NF-κB were showing up. So to follow up on this, we then looked at transcription factors that are differentially open or active across all of the different states, because transcription, assuming that these cell states are regulated epigenetically, transcription factors will be the key regulators. So again, there, subunits of AP1 and NF-κB were showing up as the top key regulators for these regenerative cell states. And then we also confirmed that by correlating the expression with the activity of these transcription factor motifs, especially for subunits of AP1, as you can see all here at the top right corner, showing the highest correlation. What was interesting here is also this intermediate state that we basically didn't know how to call, and we just called it like this because it was sitting between those two cell states. We could see that it also shows kind of shared open motifs both with the regenerative cells and also with the stem cells, again pointing to that this is a more plastic and transitional state. We then looked at all open regions across both the ones that are differentially open in the regenerative versus the stem and the other way around. And again, we can see that the intermediate sits right between these cell states. So we then used a computational method called CMYC+, to derive gene regulatory networks. So basically using sort of a correlation of transcription factors with other genes to find potential targets, and then looking at the enrichment of transcription motifs in our ATAC-seq data. And combining all of that, we found a list of potential target genes for each of the transcription factors we were interested in. So our top regulators were, of course, AP1 and NF-κB, and what we could see is that the genes, the potential target genes, were highly upregulated in the regenerative cells compared to all the other cells. As a comparison, we always use the stem cells, and for example, what was interesting was that we could see that ACL2, which is a known stem cell transcription factor, was very high in the canonical stem cells, the LGR5 positive stem cell. However, the stem notum seemed to be regulated by LEF1, so a different transcription factor. So this was showing that there are further differences between these two cell states. We also used as a validation experimentally derived target genes of AP1 with ChIP-seq. So this was unfortunately not in colorectal cancer, it was in esophageal cancer, so another gastrointestinal. But there was a big overlap with our computationally predicted target genes, and again, those were highly upregulated in the regenerative states. So we then wanted to know if this is true in primary colorectal cancer. So up to now, of course, our generated data, the multiome data, was from liver metastasis. We didn't have, unfortunately, multiome data from primary, but we, again, found publicly available data. And even though this was a much smaller cohort of patients, again, we were able to find the same subunits of AP1 showing up as very active and open in the regenerative cells. So we then looked into a bigger cohort. This was bulk RNA and ATAC-seq data. So we found a cohort of around 200 single glands that were extracted from patients. And we separated the glands into the glands that have high signatures of the regenerative cells versus the ones with low. And then we looked at the AP1 target gene expression, and we saw that they are very much upregulated in the high rec glands. But more importantly, the AP1 motif was also enriched in the differentially open chromatin regions in the high rec glands. And so finally, we wanted also to validate this experimentally. So for that, we used patient-derived organoids. So we were able to establish organoids from 3 different patients. I'm showing here one, but the results are very similar for all of the 3. So first, we used a lentivirus with a dominant negative form of FOS, which is one of the most common subunits of AP1, that then forms a non-binding heterodimer with Jun, which is another one of the most common subunits, and that prevents AP1 from binding. However, it's shown here on the first plot, when we use the organoids grown in standard media, we call it 3D here, we saw that when we inhibited AP1, nothing happened. So we looked at, with qPCR, the expression level of marker genes for the RECs versus the stem cell, and there was no really difference. So we thought that this could be because the media is pushing the cells into stem cells, and because that's the standard media. So what we decided to use is we decided to experiment how to push the cells into a regenerative state. And there was a recent report where they managed to do this using collagen. So we then grew the organoids in 2D using collagen coated plates. And first, what we could see is that 2D versus 3D, the marker genes of the regenerative cells are much more upregulated, whereas the stem cell markers are downregulated, showing us that this is indeed, it is indeed pushing the cells into a regenerative state. So when we then inhibited AP1, we could see a marked decrease of these marker genes of the regenerative cells, followed by an increase of the stem cells, showing that the cells are transitioning into stem cells. And this was also reflected in the growth of the organoids that slowed down after inhibition of AP1. So to sum up this part, we could show that the regenerative cells, the key regulator of the regenerative cells is AP1 and NF-kappa B. We are now trying to validate NF-kappa B, whereas the stem cells are also different between the 2 subpopulations, and we think that LEF1 is the one that is important for the stem node. Sorry, it's not moving. There we go. So for the next part, we wanted to look more in the tumor microenvironment and the role of the tumor microenvironment in driving or maintaining these cell states. For this, we turned into spatial transcriptomics. We used the 10x Genomics Visium technology. So basically, we have a tissue section which we place on a slide, which has these capture areas. These capture areas has 5 to 55 microns, so it's not on a single cell resolution. We usually have from 1 to 15 on average cells on each slide. So to be able to, where we get the expression, we usually get around 10,000 transcript per spot. So to be able to estimate what we have in each spot, we use deconvolution. So we use our single cell data, and then we map it onto the spatial data, and we estimate for in each spot the percentage or the abundance of each cell type and cell states. And then we look at the cells that co-occur together more often than by chance, and we cluster these spots, and we are able to infer then cellular neighborhoods or niches where cells are co-occurring very often. So starting first with primary colorectal cancers, we looked at some samples that were annotated by pathologists into tumor core versus the invasive edge. And when we mapped our cell states, both cancer cell states and also the tumor microenvironment, what we could see is that, interestingly, the regenerative cells are very much enriched at the invasive edge compared to the stem cells that are all here in the core, marked with green and blue. But even more interestingly, these cells, regenerative cells in the invasive edge, are residing in immunosuppressive niche that consists of different types of tumor-associated macrophages. For example, SPP1 macrophages that have been shown in the field to be very proteogenic and prometastatic. Also neutrophils, which don't seem to infiltrate the tumor where the stem cells are. And also they are surrounded by cancer-associated fibroblasts, specifically extracellular matrix remodeling CAFs and myofibroblasts, showing us that these cells have a potentially important role in maybe maintaining or sustaining this regenerative phenotype. So when we then clustered these spots, we inferred cellular niches. We could see that this kind of invasive immunosuppressive niche nicely clustered into one niche or one cluster, number 4 here, and then we could show statistically that these cells are enriched into this specific niche. So then we looked at the genes that are upregulated in the invasive edge versus the core. And what was, again, interesting here is that in addition to the pathways that we were expecting, we also, again, saw MAP kinase signaling. And we saw the same when we spatially mapped the signature of MAP kinase signaling compared to, for example, Wnt signaling. As expected, we see it in the core where the stem cells are. So we then turned to a TCGA cohort because it's a large cohort of samples. It is bulk data, but we used the proteomic data where we classified the samples into samples with high REC signature and samples with low REC signature, and then we looked at differential expression analysis. And again, we saw components of the MAP kinase pathway showing up very clearly in the ones with high REC signatures. And then we used our organoids again. So we compared 2D versus 3D, because 2D is enriched with the regenerative cells, 3D is enriched with stem cells, and we could see that in 2D, these cells are very much more sensitive to MAP kinase inhibitor, trametinib, showing us that MAP kinase signaling is associated with these regenerative cells, and also this could be a potential therapeutic sensitivity for this cell state that from our other preliminary data shows that it's really resistant to chemotherapy. So we then turned to liver metastasis and to look at the spatial organization of the cells in liver metastasis. The analyses were more complicated there because there was a lot of heterogeneity across the samples, and part of this was because there were different histological growth patterns in liver metastasis depending on how the cancer cells interact with the liver parenchyma. So the two most common growth patterns are desmoplastic growth pattern, where the tumor cells are separated from the liver side with this desmoplastic rim that is composed of fibroblasts, cancer-associated fibroblasts, and ECM molecules. Whereas in the replacement growth pattern, the tumor cells come into direct contact with the liver, and they start to kind of replace the hepatocytes, and they take over the sinusoidal vessels, so they don't basically need angiogenesis. And then it has been shown that this is clinically relevant because it has been shown that this replacement growth pattern has worse prognosis and worse response to therapy. So we were able to find samples with different types of patterns to compare. And again, with the same computational platform, we mapped the single cell data onto the spatial data, because this is, again, Visium data with spots that have 1 to 15 cells. And what we could first see is that in the desmoplastic growth pattern, there is a really nice rim here composed of fibroblasts. In our case, this was specifically immunomodulatory fibroblasts and ECM also CAFs. And we could see that a lot of the immune cells are also on this border, and they don't really infiltrate. And the only ones that do infiltrate are the immunosuppressive neutrophils and the SPP1 macrophages. But looking closely at the distribution of the cancer cells with the immune cells, even though it's not as clear as it was in the primary colorectal cancer, we could see again the inflammatory RECs, so not all the RECs now, but just inflammatory RECs sitting very close by these SPP1 macrophages, neutrophils, and CAFs. It's much more clear in the replacement growth pattern, as you can see here, for example, and here, that they again sit in that same immunosuppressive niche. So then we derived this computationally. We again looked at cellular neighborhoods, we clustered the spots, and what we could see is now we have two important neighborhoods, important for us, of course, where we have this IREX, so just inflammatory regenerative cells. One is more prominent in the desmoplastic pattern and the other one in the replacement pattern, but they both show co-localization of these inflammatory REX near these immunosuppressive cells. So confirming that they recreate the same immunosuppressive niche in distant metastasis. So then this enabled us to look more into cell-cell communication, basically looking at which ligands coming from the tumor microenvironment are predicted to mediate this inflammatory regenerative phenotype. So to do this, we used a combination of two methods. First, we looked at the enriched ligand receptor interaction using the method we developed called CellPhoneDB, but that gives you a bigger list of potential interesting interactions. So to shorten that, we focused, because we also had attack-seq data where we can look at the gene regulatory networks, we wanted to predict which of those ligands are the ones that activate downstream signaling in the responsive cell, which is the cancer cell, and specifically activate the EP1 target genes. So we were able to shorten this list, and we are now focusing, so we were able to identify a few growth factors and pro-inflammatory ligands that we're now focusing on more in detail, and we're trying to validate this in the lab using organoids. Reassuringly, we found that when we looked at the spatial enrichment of these ligands, these two plots on the left show this in two different samples, but this was repeated across all samples we had. Most of these ligands are also spatially enriched in that specific neighborhood compared to all other neighborhoods and compared to all other genes. And we then looked at, again, at the TCGA data, even though, of course, it's bulk data. We use correlation now with these ligands, with the signatures of regenerative cells compared to stem cells, as we could see that a lot of these ligands have a very high correlation, especially the inflammatory regenerative cells seem to be very, their phenotype seems to be very tied to specific cells in the tumor microenvironment. So just to summarize, what I have shown today is that we used single-cell multiomics and spatial transcriptomics data to characterize the different phenotypic cancer states, both in primary and liver colorectal cancer. What we've identified is these regenerative cells that seem to reside in an immunosuppressive niche at the invasive site of primary colorectal cancer. We think that there is an intermediate state that is more plastic and that transitions into a stem cell, and that they manage to recreate the same hierarchy at the liver site as well as the same immunosuppressive niche. And we've also found that AP1 is a key regulatory factor for these cells. And I'd like to thank my lab. This was work by Sam and Nasrin, our tissue bank, our collaborators, and also the patients and their families for their very generous contribution. And thank you very much for your attention.
Thank you. Okay, so now we have some time for questions, so please approach the microphones. And also I remind those people connected virtually that they can ask through the chat. So, first question, please.
Hello, I'm Anna Vähärautio from University of Helsinki, and really nice talk, first of all. So how do you see the pattern here that you link to metastasis, I understood especially? How does it link to resistance to treatment? Because I study ovarian cancer, and there we see a very similar pattern from many different methodologies, somehow linked to resistance and sort of selection through chemotherapy.
So I haven't shown this because it's quite preliminary, but what we have done is we've used both our data to compare treated versus non-treated, but also we looked at organoids where we treated them. And immediately after treatment, they become very regenerative like this. So And then after they revert back, but they don't fully revert back to what they were before. And we think they become more stem notum instead of stem, and we think that leads to more non-canonical states. They've been described in a very recent publication in very rare patients that they start to become more squamous-like, and we start to see that in the organoids. So we think that actually chemotherapy, it's very early to say, but potentially makes them more pro-metastatic because they don't seem to revert back to the original. They regrow, but they seem to become worse. But it's very early, that's why I haven't presented. It's very early to say.
Yeah, we see both in kind of organized system and in the clinical tumors, this sort of similar AP1 NF-κB
really?
state.
In ovarian?
Yeah, you should come to my talk tomorrow. Actually,
okay, yeah, that's
it's really sort of pointing to the same direction. And the other point was that now that this is sort of an anti-stemness state, how do you see, how does one study this in the organoid context, and how does this sort of the pro-stemness, let's say, context we need to provide for the organoids, how does that kind of deviate our results, for example, related to resistance or different sort of phenotypes we want to study in ex vivo system?
Yes, it's been a problem for us because they do look more stem-like. And we are still experimenting with a larger number because so far we've profiled only 2, so it's very early to say. But they are different, the 2 of them. One is more stem, the other one is less stem, even with the same media. But I guess it would depend on what kind of mutations they have, right? Because once they get APC, of course, they are Wnt independent. And once they get KRAS maybe or something, so it depends. We haven't profiled their mutations. That will influence, I think. But it is a problem, yes, because the media is pushing them more to be stem. And so that's why we're experimenting with all of these ligands. Because we do have a way to get them into regenerative, but it's 2D. When they're not 3D anymore, so we are trying to find an easier way to push them in so that we can then perturb them again. Yeah, I think the media is not ideal, unfortunately, from what we've seen. Yeah.
Yeah, in ovarian cancer, it's not even sort of pro-Wnt signaling media actually, but still we see sort of similar features and the difference between the tumors and the organoids. Yeah, thanks.
That's very interesting. Thank you.
Another question there.
Hello, thank you for the presentation. And about the metastasis in liver, the pattern of the mutations and the expression profile have been demonstrated to be similar to the original tumor, or sometimes are different?
So in our patients, we saw very similar expression profiles. And as I mentioned, there was a very recent publication where they show some very non-canonical states, like the squamous, which is really, you can't find that in the normal intestine. And we didn't see it in ours, but we had a lot of samples that were not treated. We were lucky to get a lot of samples that were not treated. We think it's probably due to therapy, because when we integrated our data with a lot of other publicly available, I haven't shown that, but when we increase the number of samples, we do see that kind of non-canonical squamous state only in 2 patients, let's say out of, I think it was 26, 27 or something like this. So there is something happening differently in metastasis. And to me, it seems like it's due to therapy because it's only in treated, but it's very difficult to say from the number of samples. We don't have enough to prove that. I think there is something happening different. Maybe just in our samples, we didn't detect it, unfortunately, because a lot of them were untreated.
Thank you.
Thank you.
So your work is based on metastasis in liver metastasis. What is it known about metastasis in other organs? For example, when you think about the bone or lung or...
So there are often metastasis. The liver is the most common, but there are lung and mental metastasis, but we haven't been able to get samples. It's been on my, I've been trying to, but I think the person we collaborate with is a liver surgeon, and it's quite difficult to, I think they're rare, it would be very good to compare them, but it's
Would you expect the same, I mean, I don't know, you can speculate, what do you expect to find?
I would expect, because we see the same, I would expect definitely to see the same, potentially something new like this squamous state, but I would expect at least to see the ones we see, and potentially something new. It's rare, unfortunately.
I don't know if there are more questions. If not, I think we are going to close the session. So I want to thank the speakers for fantastic talks and the audience for the discussion. So,